Okay, guys, good morning. Let's begin. Today we're going to be discussing the first step that is beyond the cell. We have been discussing the cellular architecture, the cellular structure and function. We have been discussing the nucleus, the cytoplasm, and the cell membrane and all the constituents of the cell membrane, the constituents of the cytoplasm, constituents of the nucleus. Then we talked about the interrelationship between all of them, what's happening in the nucleus, and how even before we discussed the cell, we discussed all of the subatomic particles, the atoms, the micromolecules, how they arise and become macromolecules, and how there's energy stored in these micromolecules, these monosaccharides, these disaccharides, these amino acids, and glycerol and free fatty acids, and nucleotides with these phosphates. They are all micromolecules, and then they aggregate, they summate, they add more bonds, and these bonds, they represent energy. So when your body wants energy, it will break down these bonds. And if your body wants to save energy, it will create bond. So we learned that metabolism is both. It's anabolism, where we build up the bonds, we store the energy, like we put the clothes in the closet. When we want to store the clothes and we want to use them, we go to the closet, we take them and we put them on. We saw this with the ATP. The ATP is 2-in-1. It supplies energy. When we need energy, we break the bonds. We go from triphosphate to diphosphate to monophosphate. Then remove that one phosphate. We go back to a nucleoside. And then if we have used all the energy and we no longer want to use the energy, we're not running no marathon, we're not consuming no energy, then we're going to be storing that. So we're going to go from a nucleoside to adenine monophosphate to adenine diphosphate to adenine triphosphate. Same thing with the sugars. The sugars, we begin with glucose, then we add another glucose to it. We get a disaccharide, maltose, then we make a polysaccharide. And if we want energy, we break down. We break down the polysaccharides down to the disaccharides and monosaccharides. So that movement, that shifting, going upward, building up anabolism, breaking down, going down, catabolism, freeing the energy, exergonic reactions when I need energy, endergonic reactions when I'm storing the energy. That is metabolism. Then we went up and we discussed how the cell regulates that metabolism. It's by the enzymes. What are enzymes? Enzymes are proteins. How do we get enzymes? We have to go inside the nucleus, transcribe mRNA using an RNA polymerase. Then we get our nascent mRNA transcript. And that nascent mRNA transcript must undergo post-transcriptional modification. We chop out the introns and we stick together, we glue together the exons. And then the exons are going to get translated in the ribosome, and then we get our proteins. Proteins, they could be protein channels. Protein channels allow for the passage of polar electrolytes through the nonpolar plasma membrane. The enzymes, they accelerate rates. These are biochemical pathway catalysts. They catalyze, these are biocatalysts. They catalyze the rate of the reactions. So all of that is happening in the cell. So now we're done with the cell. We now learned about the subatomic particles, the subcellular particles. We talked about the organelles. We talked about what's happening in the cell. Now we're going to move up. We're going to learn that these cells, although the cell is the basic unit of life and the basic unit of health, the basic unit of disease, also the basic unit of death, this cell does not work on its own. The cell is actually very tightly connected to nearby cells. We talked about that, about these CAMs or caps. CAMs are cell adhesion molecules, and caps are cell adhesion proteins. They are all sticking together. These are gluing the cells together. So if you guys are, for an example, cells, you guys are separated. You are not attached to each other. So the cells, imagine you are like in an auditorium. You're so like in the subway. You are sitting so close to one another. So cells are really tightly held together by cell adhesion proteins, CAPs, and cell adhesion molecules, CAMs. And there's tight junction, there's gap junctions. The cells have holes between them, but these are not just holes, these are protein complexes known as connexins, like donuts. These are like between the two cells that allow for the passage of solutes, nutrients, ions, and their cytoplasm is just so connected. We talked about this, about how I told you there was a neighborhood that I went into, and all the homes, they have like doors where you can just go through the home and go access a particular shared part. So all the homes, there's a public part of the home and there's a private part of the home. So if you can go through, like for example, if the houses are connected, people can only publicly walk in the street. You can never enter the house and be in a public part of the house. So the house is private. So your house is private. Okay, I can never enter it unless I am visiting you. The street is public, it's for everybody. So I went into a place where the homes are, so the street is public, but the homes is divided. Some part is private and some part is like the street. So the cell is very similar to that. The cells are connected in the sense that the extracellular compartment in the interstitial fluid is public. All the solutes are there. But there are regions inside the cell where the cells are connected with these connexins, as we will see today, that allow for the passage of solutes to a certain part that allows the transmission of electrolytes, transmission of electrical stimulation. And then there are regions in the cell that are very private to the cell. These are regulated by hormones and transcription. Does that make sense? Okay, so now we're going to learn that these cells, obviously, although they all have the DNA, the same DNA, they do not look the same. And they discovered that when they brought one cell, skeletal muscle cell from the frog, skeletal muscle cell. They brought one skeletal muscle cell from a frog. Let's assume that this blue marker is representing a skeletal muscle from the frog skeletal muscle. That's the nucleus of the frog skeletal muscle. And this is, let's say that this is the ovum, the egg cell of the frog with its orange nucleus. That's the frog ovum. So what they did was they removed the nucleus of the frog ovum. So now the frog ovum has everything that the cell has except the nucleus. And they removed the nucleus of the skeletal muscle. And they put the nucleus of the skeletal muscle inside the frog. And they put the nucleus of the frog ovum inside the skeletal muscle. Does that make sense? Does that make sense? What happened here? And then they put them in their function. What does the ovum do? It gets fertilized by a sperm. It gets fertilized by a sperm, and it then must divide. Must divide after the union of the sperm. This is high school. The union of the sperm and an egg, and then the cell division, the blastula, the morula, the gastrulation, and then you have an organism. What does the skeletal muscle cell do? It must be able to transcribe proteins that are cytoskeletal proteins, like microtubules that help it, contract and be able to lift stuff so strong. So they said, let's see what's going to happen. The hypothesis was the cell is going to die because now you're going to have a new nucleus in there. Guess what happened? So now they put it in its medium. They fertilized this frog ovum with a sperm. Everything went perfectly fine. The cell behaved as if its nucleus was not changed, as if this was the orange nucleus. It wasn't blue. It wasn't from a skeletal muscle cell. It wasn't like the original one was there. And when they put this one back into the tissue, it divided and it had the same structures inside that are cytoskeletal. It looked just like all the cells, although the nucleus was from the ovum. So what does that tell you? That tells you that the DNA is the same. If you give $1,000 to a police officer, $1,000 to a makeup artist, $1,000 to an athlete, $1,000 to a chef, the chef is going to take the same $1,000, buy the cooking utensils. The makeup artist, she's going to go and get all of the makeup stuff to help her do her job. The police officer, for example, is going to go buy weapons and, taser, and the athlete is going to go buy the sneakers. So it wasn't $1,000. It was who you are that led you to spend that money in the way that helps you accomplish your function. So that's the skeletal muscle cell. So the DNA is there, but they have the set of the polymerases. Polymerase, remember the polymerase? Who goes when we go from DNA to mRNA? That is trans. What is that called when we go from DNA to mRNA? That's transcription. Then we translate mRNA into our proteins that make the cell who it is, and that is known as translation. Who are the enzymes that take us from DNA to mRNA? Who are the enzymes that mediate this transcription? What are they called? I did that last week there. How did we go from DNA to mRNA? Who's the enzyme? Who's the enzyme that mediates that? Nobody in the class knows. What is the name of the enzyme that mediates the transcription? I'm sorry? RNA polymerase. Because the polymerizes the mRNA, and we said that because it makes RNA, that makes it a RNA polymerase. What did it depend on? The DNA. So it's a DNA dependent RNA polymerase. So in the skeletal muscle cell, do you have the same RNA polymerase? No, you have a skeletal muscle DNA dependent RNA polymerase. In the ovum, you have an orange ovum DNA dependent RNA polymerase. So when we go to the DNA, there is a region for all the protein that the skeletal muscle cell needs, and a separate region that the ovum cell needs. So when they replace the nucleus of the skeletal muscle and put it in the frog ovum DNA dependent RNA polymerase said no problem, even though it's a different nucleus, the DNA is the same. It's the same $1,000. So I'm still gonna go to this region right here. The other cell, the frog cell, the muscle cell, they had their muscle RNA dependent, they had their muscle DNA dependent RNA polymerase that went to the skeletal muscle region and transcribed for us the proteins to make us skeletal muscle. So that's how these cells aggregate together, and that's how they now make a tissue. So now from skeletal muscle cell, you're gonna have skeletal muscle tissue. And from this frog ovum, you're going to have a fertilization that's going to enable and facilitate for the development of a fetus. You're going to have the blastula, the gastrulation, and then you're going to have the development of this fertilized fetus. So now we're going to see that we're going to start seeing that tissue in our body are dependent on the same forms of cells aggregating together, but not just that, also associating with other various types of cells. So, for example, we have the epithelial tissue. That is the baseline tissue for our skin. But does that mean our skin has no nervous tissue? Otherwise, you will not feel a needle when you get a needle injection. So there is nerve tissue associated with your epithelial tissue. Yes. The RNA polymerase is an enzyme, and it's an enzyme that is responsible for polymerizing this macromolecule known as the mRNA. And the DNA is the transcript that we use to transcribe that mRNA. The nucleolus is responsible for having regions of the chromosome that are just specific for rRNA, ribosomal RNA. So the polymerases are there inside the cell and they know where to go and where to transcribe based on what cell are they in. So the RNA polymerase in a skeletal muscle cell, an RNA polymerase in a neuron, and an RNA polymerase in a hepatocyte. They're not going to go to the same region. They're going to go to certain regions that the same way the athlete will go to Foot Locker to shop, the cop will go to the gun store to shop, and then the chef will go to buy their utensils from a different place. The same thing. The polymerases, they will not go to the same place in the DNA. The DNA is so vast and so big. And we said in the beginning of the course that there is a euchromatin. That region of the DNA is the region responsible for, upon being transcribed, it will give the mRNA that will later be translated to give you the protein that make you who you are. It is the proteins that make you what kind of cell you are. But you're never gonna get the proteins unless you get a RNA. And you're never gonna get RNA unless you transcribe DNA. So the DNA is the most important. But itself on its own, it cannot do anything because it's just laying there. You still need a team of polymerases that will go, these polymerases will go and they will be able to these polymerases will go and they will transcribe the regions of the DNA that give the cell its identity. Yes, I'm sorry. Yeah, it's already posted. Okay, I'll show you in the end of the lecture where it is. Okay, but that study guide that I gave you, like I said in the beginning of the course, any textbook, these are there. You see, now these lecture slides, these were from another professor, and she did not use patent. It doesn't matter. What matters is any book or any set of PowerPoint slides that I post for you. You can utilize those to solve your study guide. You can utilize online resources to solve your study guide. You're not to be overwhelmed by all of this material. You are just responsible for that study guide and those homework questions. So two things, the homework questions and the study guide. You have to answer them and you have to be able to really understand them. And then when the night before the exam, you're going to submit it to the portal. Okay? All right. So now histology is a branch of anatomy that represents dissecting and studying the architecture of anatomical figures that cannot be seen with the naked eye. So back in the day, anatomy and physiology was all about cadavers, what you can see with your eye. Fixating, using a fixative histological medium where you add formalin, and that formalin prevents the putrefaction and prevents the decay of your anatomical figure that you would like to analyze. But then they realized that anatomy does not begin at the level of the naked eye, your organs and your organ systems. Actually, when the microscope came in, it gave us a whole different field that is below the naked eye, and that is known as the microscopic anatomy or the histology. Okay, so the tissue is comprised of group of cells that work together to perform a specific function, and these cells are similar in structure and function, and they cooperate to carry out specialized tasks within the body. So the principal types and development of the tissues. There are four types of tissue. There's the epithelial tissue, and this epithelial tissue is very interesting because it is bound to the basement membrane. It is bound to the basement membrane, but it is bound in a basement membrane in a very interesting way. You have cells that you have, for example, simple epithelium. Simple epithelium is made up of cells that are 100%, every single cell is attached to the basement membrane. But you have other types of cells that are epithelial cells, and these are known as stratified. So these are not simple epithelium, these are stratified epithelium. Stratified epithelium are examples of cells like if you are in a stadium, you have one layer of the stadium that is on the ground, then you have the other layers are on top of each other in the back. That's exactly how the stratified epithelium looks like. You have one subset of tissue layer that is attached to the basement membrane, while the remaining cells are laying on top of each other. So that's different from simple epithelium. We will see that. Connective tissue is an example of tissue that's mainly responsible for, from its name, connecting. So it connects the epithelial tissue to the basement membrane. It connects nutrients and delivers them to muscular tissue and to nervous tissue. So this is a mediator. The connective tissue is mediating mechanical support. It's mediating the delivery of metabolic advantage. While muscle and muscular tissue, nervous tissue, those two, even though both of them are different types of tissues, they are actually very similar. And that both are excitable. So epithelial tissue is not excitable, and connective tissue is not excitable. But muscle and nervous tissue both are excitable. But there has to be something different. That's why they have two names. If they were the same thing, you would not have two different types of tissue. So you must understand that the muscle tissue and the nervous tissue, both of them are excitable, meaning membrane voltage change due to the opening of these ion channels. We said ion channels can open because of a ligand. These are known as ligand-gated ion channels. Also, there are ion channels that are open because of heat. When it's so hot, they open. Other channels are open because of mechanoreceptor. Okay, these cells open and they allow for the passage of electrolytes. The passage of electrolytes changes the voltage. When the voltage changes, now the membrane of the nervous cell is able to relay and mediate the passage of a stimulus. The same thing with the muscle tissue. But when the muscle tissue is excited, it does not propagate or mediate the passage of a stimulus or an input or an output. That's the job of the nervous tissue. It is the muscle tissue that will contract. So when you excite nervous tissue, it will allow for the passage of input, stimulus, input information, or propagate and output information for integration, for homeostasis. getting the information to pass through to an integrator and receiving the information from an integrator. That is the job of a nervous tissue once it's stimulated. It's stimulated and excited. That's why they call it excitable tissue. If there's no excitation, it will never do its job. So both of them are excited. The nervous tissue is excited, the muscular tissue is excited, but it's only the nervous tissue that once excited, it will relay input signal and output signal. But when the muscle tissue is excited, it will not relay no information. It will contract, get shorter, but still based on excitation. So epithelial tissue is a type of tissue that covers the body surfaces, and these body surfaces like your skin, and the body surfaces are your skin and the inner cavities. Like for example, we are hollow from outside and from inside. So the food that made it to your digestive system, but it never got absorbed. It never went from your intestine into the blood. That food has actually never been inside your body, even though it actually was. You swallowed the food. But if you swallowed it and vomited it, that food was still outside. It's only inside your body when it was absorbed into the blood. So getting food in your belly, that does not mean the food is you had a meal. No, because you have to ensure that the meal that you had, you were able to absorb it. Just because money was deposited in your bank account, it does not mean you have it, because you need to be able to withdraw it. Then when you withdraw it, then you have it. It's still outside. Even though it's in the bank, it's still outside. Same thing with the food. Even though it made it to the inside of you, and it's stored in your intestine, but it didn't make its way through because it wasn't fully absorbed. That's why we are hollow from outside and we are hollow from inside. And who lines the outside? It is the tissue that also lines the inside, and that is your epithelial tissue. So you have to know for your NCLEX, for your exam, that what kind of tissue lines the outside, what kind of tissue lines the inside, and that is known as epithelial tissue. Excellent. Epithelial tissue is a type of tissue that covers the body surfaces and it lines the body cavities and forms the glands. It consists of closely packed cells arranged in one or more layers, and the epithelial tissue serves as a protective barrier. It controls the permeability, and it is involved in the secretion, absorption, and sensory reception. So secretion is, there is secretion and there is excretion. Excretion is the useless substance that is harmful, toxic, and we need to get it out of our body. And the secretions are the useful things that come out of the gland, like the exocrine gland and the endocrine gland. Your pancreas secretes hormones, insulin, glucagon into the blood. That's a secretion. It secretes digestive enzymes like trypsin, like chymotrypsin, into a duct inside the intestine to help the digestion and breakdown of food, enable to absorb it into your blood. So that is all epithelial tissue. That is all epithelial tissue. Epithelial tissue is important for protection because it shields the underlying tissues from mechanical and chemical damage. And that is part of your innate immunity. When you learn the innate immune system, the frontier, the first line of defense is your skin. It's like in a country, the first army that fights the invaders is not actually the police and the army and the navy and the air force. It's the mountains that are outside on the borders. Okay, they prevent the passage of these foreigner invaders to come through. So that these mountains, this fence. So your house is surrounded by a fence to protect all the intruders, to protect you from all the intruders who are trying to come in. So the epithelial tissue is the one that protects you. Absorption. Absorption allows for the uptake of substances such as nutrients in the intestine, And it also important for the secretion and excretion, produces and release substances like enzymes, hormones. We talked about that, from endocrine glands and exocrine glands. Sensation contains, it means that it contains sensory receptors for detecting environmental changes. Connective tissue now is a diverse group of tissues that provide support. They bind together and they protect the tissues and organs of the body. They are characterized by a large amount of extracellular matrix, which includes fibers, collagen, elastic and reticular fibers, and ground substance. So this connective tissue is important because, as we said, it mediates, sticks together, glues together, and surrounds the tissue, protects its integrity. And we have different types of tissues. You have tissues that connect bone to bone, like a ligament. That's connective tissue. You have tissue that connects muscle to bone. That's a tendon. That's connective tissue. The blood is a connective tissue. This one, you will see it on your NCLEX. What kind of tissue is your blood? Is it epithelial tissue? Is it nervous tissue? Is it muscular tissue? No, it is connective tissue. Blood is connective tissue. So there are different types of connective tissue structurally, and therefore they will have a different function. So there's loose connective tissue that supports and binds other tissues, like the areolar tissue. There's dense connective tissue that provides strength, elasticity for the tendons and the ligaments. And there's cartilage. Cartilage cushions the joints and supports the soft tissue. Bone provides structural support and protection for the organs, and the blood transports nutrients, gases, and waste products. Muscle tissue. Definition of the muscle tissue is that it is a specialized tissue that is capable of contracting and generating force to produce the movement. It is composed of muscle cells or fibers that contain contractile. You see? So now once they get excited, they contract. But remember, they will never contract unless they have been excited first, such as actin and myosin. So there are different types of muscles. There is skeletal muscle that is like in your biceps and your triceps, and that's important for tensile integrity. And they are attached to the bones, and they are responsible for voluntary movement. Cardiac muscle is found in the heart, and it is responsible for pumping blood through the circulatory system. Smooth muscle is found in the walls of the hollow organs and the intestines, in the blood vessels, and is responsible for involuntary movement. Like the smooth muscle lines your blood vessels. It's very important for you to make the connection of where do we find skeletal muscles. These are on your bones, surrounding the bones, and they mediate movement, lifting, locomotion, the ability to move from one area to the other. And then there is cardiac muscle. The cardiac muscle is important for the pump, the heart pump that pumps blood, nourishing the tissue with oxygen for oxidative phosphorylation, for aerobic respiration, providing nutrients to the tissue and removing metabolic waste and toxic waste byproducts. That's your cardiac muscle. And the smooth muscle, it's a muscle, but it's not cardiac, it is not skeletal, it's smooth muscle. But remember, when you are a muscle, whether you are skeletal muscle, whether you are cardiac muscle, whether you are smooth muscle, you will contract. So the smooth muscle in the blood vessel contract, that's vasoconstriction. They allow for less passage of blood, and that increases the blood pressure. Now you have to pump harder. So when people have a low blood pressure, they have hypotension, they are given for example, phenylephrine, and that will constrict their blood vessels and stimulate the heart to pump more, increasing the blood pressure. And if the blood pressure is too wide now due to some form of a shock, like a septic shock or some immunological reaction that causes excessive vasodilation of the blood vessels, then we need to constrict. We need to constrict those blood vessels to restore the heart rate to go up. So we mediate this control of cardiac output with smooth muscles, not just cardiac muscles, smooth muscles who contain the blood and pass the blood. These are responsible for contracting and relaxing based on excitation. So once they get excited, they contract, and once they contract, that's known as vasoconstriction. And when they relax, that's known as a vasodilation. They constrict, allowing less passage of blood, increasing blood pressure, and they relax, allowing for accommodating more blood through them, allowing more blood to pass through, and that is known as vasodilation, and that actually reduces the blood pressure. But all of them, muscles, all of them contract because they are all excitable. Now, nervous tissue. The nervous tissue, the definition of the nervous tissue is that it is a specialized tissue that is involved in the reception, the transmission, and the processing of the information. So once it gets excited, once the nervous tissue gets excited, it will not contract. It will receive the information, integrate it, process it, transmit it, and create a form of an output. The nervous tissue consists of neurons which generate and conduct nerve impulses, and they have also glial cells which support and protect the neuron. So in your nervous system, you have these nervous cells that are able to receive neurological input, integrate it, transmit it, and generate an output. But there are other types of cells in your nervous system whose job is just to support the main parenchyma. And you will see this in all the different systems in your body. So for example, in your digestive system, you do have organs that are directly responsible for receiving food, chopping up food, digesting food, ingesting the food, absorbing the food, excreting the food. But there are also organs who are still part of your digestive system, but they are not directly involved in digestion. They are just there to support the digestive system. And I mentioned that, like your pancreas. The pancreas does not digest the food itself, but it supports, like the gallbladder does not digest the food, but it supports your digestive system. Here, your neurons are different from the neuroglia. These neurons are directly involved as computers. They receive information, input, integrate it, generate an output. But the neuroglia, they cannot receive input or generate an output. They are there for structural and functional support of these neurons. So the nervous tissue functions are, it's important for sensory input. It detects changes in the internal and external environment. So we talked about this in the first day of class about how, in sensory input, we receive that hyperthermia or hypothermia, hypertension or hypotension, hyperglycemia or hypoglycemia, any hypo, we don't want it, we want U. But once this U state, this U homeostatic healthy range has been departed either to hyper or hypo, there are sensors who will sense that deviation, and they will send input signals that will integrate the information, and they process the sensory information and determine the appropriate response. Then there's a motor output. The motor output will transmit the signals to muscles and the glands to elicit a response. Homeostasis. Homeostasis helps to regulate and maintain stable internal conditions. We now have to talk about the embryonic development of the germ layers. So there are germ layers that start folding. They start folding immediately after the process of fertilization, the process of generating this new fetus, the blastula, the gastrulation, the development of this fetus that will later on become a toddler and then become an early adolescent and then grow up into adulthood until it reaches the old age of senescence. That all begins, all of these tissues have three origins. And the three origins are the ectoderm and the mesoderm in the middle. And remember that the mesoderm always gives rise to the mesenchyme. The mesenchyme is a special type of tissue that arises not from the ectoderm, not from the endoderm, but rather from the middle layer known as the mesoderm. So the endoderm is the innermost layer of the three primary germ layers in the early embryo, giving rise to the epithelial lining of the digestive tract and its associated organs such as the liver and the pancreas. The mesoderm is the middle of the three primary germ layers in the early embryo, which develops into muscles, bones, blood, and in the urogenital system. That is your mesoderm. That's the middle layer. The endoderm is the innermost of the three primary germ layers, and it gives rise to the epithelial lining and the digestive tract. We talked about that the outside lining and the inside lining of our hollow organs is always by our epithelial tissue. And where does our epithelial tissue arise from in our primary germ layers? It arises from the ectoderm, the outside layer, ectoderm. The mesoderm is the middle layer. It is the middle of the three primary germ layers in the early embryo, which develops into muscles, bones, blood, and the urogenital system. The ectoderm, or the outermost of the three primary germ layers in the early embryo, forming the nervous system, including the brain and the spinal cord, the skin and the hair. So the ectoderm is the outer layer, is the outermost layer, and is responsible for the genesis of the nervous system. Now, the extracellular matrix. The extracellular matrix is a jelly-like fluid that is between the cells, and the extracellular matrix is a network of proteins and carbohydrates, like glycoproteins and glycosaminoglycans. These proteins are associated with sugars, glycoproteins, glycolipids that are there for connection between the cells that are adjacent to one another in the tissue. The particular makeup of the extracellular matrix is the extracellular matrix composition varies based on the tissue and the type. So there is a different variation, different amount, depending on, are we dealing with muscular tissue? Are we dealing with nervous tissue or epithelial tissue or connective tissue? The amount of extracellular matrix between the cells is going to be dependent on their function. So muscular tissue does not have a very thick extracellular matrix because these cells have to be very close to one another, working like a syncytium and being able to contract in order to pump blood. However, cells that are not working in a mechanical fashion, like the bone cells, those they have to have really strong extracellular matrix because that extracellular matrix associates with the bone tissue, for example, providing the bone tissue with salt, phosphate salts, and these calcium salts that give the bone its integrity and helps it do its function for structural support and for mineral homeostasis. So the extracellular matrix is there, but depending what kind of tissue are we looking at. In some tissues, it is so thin because we don't need to separate the cells as much. But in other tissues, it is very thick, very thick, as in the bone tissue. There are varying amounts of extracellular matrix in different tissues. So different tissues have different extracellular matrix densities to suit their needs. So we talked about the extracellular matrix, and that represents the glycocalyx and the collagen fibers. And as we said, the proteoglycans and the glycosaminoglycans. These represent for me an extracellular environment rich in solutes, rich in proteins and carbohydrates and lipids. And these are the tissue layer that surrounds our cells, that give our cells the support. That is the extracellular matrix. What are the components of the extracellular matrix? Obviously, we have proteins, and these are biological molecules made up of amino acids. We went through them with the alpha carbon and these four terminals. And the proteins are essential for the structure, function, and regulation of the body cells, tissues, and organs. Structural proteins are proteins that provide support and shape to the cells and the tissues. Examples of structural proteins are collagen and keratin. For example, we said keratin is very important for the hair and for the nails, while collagen is important for the ligaments. Elastin is also important because it gives elasticity to the tissue, making the tissue more malleable. Um, we find a lot of elastin in the muscle cells because these muscle cells need to have the property of contracting, being able to fold up. So collagen is found in rigid tissues like the bone, while elastin is found in tissues like the blood vessels. What enables the blood vessel, which is smooth muscle, to contract and relax? That is the protein elastin that makes them elastic so they can accommodate an increasing, varying amount of blood depending on the stimulus and the hormones that makes them contract or relax. What are the components of the extracellular matrix? So we have glycoproteins and we have also lipoproteins. These are proteins with attached carbohydrate molecules, and they are often involved in cell-to-cell interactions and signaling. Examples of glycoproteins are fibronectin and laminin, and these are specific glycoproteins found in the extracellular matrix, and they are involved in cell adhesion and migration. Okay, components of the extracellular matrix. We have the proteoglycans, which are proteins with attached glycosaminoglycan chains, and these are important for maintaining the structural integrity of the extracellular matrix and regulating the cell behavior. What are examples of proteoglycans? These are chondroitin sulfate and heparin sulfate. And examples of proteoglycans are the chondroitin sulfate that provides cartilage with the resistance to compression, while heparin sulfate is involved in cell signaling and adhesion. So now we're talking about what are the more specific proteins that enable collagen to become very strong, making as a result bone stronger in structure than the muscle tissue. And we said that the muscle tissue is rich in elastin, that's why it's elastic. But the bone tissue is rich in this chondroitin that is made up of proteoglycans. So a good exam question would be which of the following proteins is able to give tensile strength and rigidity? Is it elastin or the proteoglycans, such as chondroitin? So proteoglycans are really strong. So we do not use them for elasticity, but we use the protein that is self-explanatory from its name, elastin, to give elasticity to tissues that must be malleable, tissue that must be elastic. Collagen. Collagen is the most abundant protein in the body and is rich in proteoglycans and glycosaminoglycans. These are very strong. They provide tensile strength to this bone structure that is rich in collagen, and it provides strength and structure to tissues such as the skin, the bone, the tendons, and the cartilage. The collagenous bone. The collagenous bone is rich in fibers that are thick. These are strong fibers made of collagen proteins, contributing to the tensile strength and the resilience of the connective tissue. Reticular fibers, these are thin branching fibers made of collagen types 3 and type 4, forming a supportive network in tissues like the spleen, the lymph node, and the liver. And collagen type 4 is a specific type of collagen primarily found in the basal lamina, which is a layer of the extracellular matrix that provides structural support to the epithelial cells. So you don't have to know all of these different types of proteins. You just need to appreciate that collagen has tensile integrity due to the presence of these different types of fibers. There's fibers, collagenous fibers, there's reticular fibers found in the spleen and collagen type 4, but that collagen is the most abundant of all the proteins in our body. Okay, holding the tissues together. So primarily the extracellular matrix is responsible for holding the tissues together, like in the skeletal muscles. And as I began discussing today in the beginning of the lecture, that these cells are held together. They're held together by the cams and the caps. Who remember what did I say the cams and the caps were? Cams. I said that cells are not sitting the way that you are sitting separated. They are like in an auditorium or like in a subway. They're touching each other. Using what? Who are the proteins that hold them together? Cams are caps. Cams and caps. What are cams? What are caps? Cams are cell adhesion molecules, and caps are cell adhesion proteins. Cell adhesion proteins. So desmosomes and tight junctions in other tissues. So desmosomes, tight junctions, and remember I mentioned the connexins and the gap junctions and the tight junctions. These are all types of hooks that hook the cells together. These are your cams and your caps that hold the cells, hook them together. In fact, they hook the cells so tightly, like a staple. They staple the cells so tightly that even water cannot leak between. Like for example, you have a lot of tight junctions in your stomach. Those tight junctions hold the stomach cells so tightly together that the hydrochloric acid cannot seep through. I mean, how tight is that? That's how strong these desmosomes are. Okay, tissue repair. Tissue repair is important because we need to be able to spin the cell division cycle, start up the cell cycle, and divide to repair tissue that was destroyed. So we do debridement, remove all the tissue that was necrotic. So if we had stress, we talked a lot. We talked about the different types of. Excuse me. Okay, guys. So we mentioned that. Okay. So we mentioned that the cell can undergo. We mentioned that the cell can undergo different forms of stress that can destabilize the cell and get it out of homeostasis. That is the cell in homeostasis. Okay, it can undergo different types of stress. Like, for example, hyperthermia or hypothermia. That is an example of physical stress. There's chemical stress, like some corrosive or detergent, corrosive chemical agent or detergent. For example, an infectious microbe such as bacteria, viruses, parasites. So they all keep attacking the cell. They attack the cell. When they attack the cell fights back. You know, you have your immune system, you have built-in adaptive mechanisms that help the cell overwhelm, help the cell win this continuous stress. But sometimes, if the onset is so severe, if the integrity is so high, if the duration of the stress is for so long, the cell will now be overwhelmed and it will be hurt and it will be damaged beyond salvage, damaged beyond repair. So the cell will now become injured. You will have cell injury, and after the cell is injured, you will now have cell death. You will now have cell death. And when the cell dies, you will have death either by apoptosis. And we said, what is apoptosis? I'm sorry? Programmed cell death. Yes. So apoptosis is programmed cell death. Programmed cell death. Programmed cell death is when the cell has actual time to kill itself, time to remove, properly dispose of itself, properly removes itself from that tissue medium, so it does not have an accumulation of damaged or harmed substance, because that would become like mad cow disease. You have misfolded proteins over many damaged cells, and that is the basis for Alzheimer's disease. There's Alzheimer's disease in the minds of many of the patients, is because the cells are no longer able to commit apoptosis. They are old, they are dead, they are rotten, but they cannot be removed and new cells can be built in their place. That's why you have Alzheimer's disease. But before, now when you're young and all of the tissue is dying, then it will be dead by apoptosis. Then we will enter the cell division cycle, the cell division, the cell division, we'll enter the cell division cycle. We will enter the cell division cycle. And in the cell division cycle, we will divide and we will make new cells. Cell division cycle, as we mentioned, is divided into two phases. The first one is the mitotic phase, but you don't get the mitotic phase unless you go through interphase. Go through interphase. And interphase is divided into three steps. The first step is the G1 phase. That is interphase. That is all interphase. It is divided into G1 phase, the S phase, and the G2 phase. Once you get, so there's a checkpoint from the G1 to the S phase, and this point, once we have the cell that has been screened and scanned, that all the proteins are healthy and all the DNA in the nucleus is ready to get doubled, then you will go from 1x DNA to 2x DNA. You double the DNA here. Then there is another checkpoint from the G2 into the mitotic phase. The mitotic phase itself is divided into prometaphase, then you have prophase, then you have metaphase, then you have anaphase, then you have telophase, then you have cytokinesis. So the cell cycle is two stages, but each stage is different, is comprised of different phases. The last checkpoint is right here, is when you go from mitosis into the G1. If you can divide again, then you will have to go through this checkpoint that will allow you to enter the G1 phase, or else you will exit the cell cycle and enter the G0 phase, known as the quiescence, quiescence. And this is a terminally differentiated stage. Like, for example, your nerve tissue is non-regenerative, cannot enter the cell cycle. Your epithelial tissue can. That's why the more you scratch your skin, more tissue is gonna enter the cell cycle, replace the one that you carved out, the one that you did debridement and removed it. But your nerve cells, once they are damaged with drinking alcohol or trauma or boxing punches to the head and all of that, once they are destroyed, they are non-regenerative, they are destroyed for life because they are in the G0 phase. They are state terminally differentiated, locked in the G0 stage. So the issue here, that is if we die by apoptosis. But if we die by necrosis, this is a dangerous form. This is a dangerous form of death. A dangerous form of death where the integrity of the tissue is going to be compromised. Because if the cell is going to be damaged is going to be killed by apoptosis, it will be broken down into small, digestible, and edible. What do we call them? Blebs. We call them blebs. These are small, edible, digestible blebs. But in necrosis, the cell membrane will break and all the content is going to leak out. When the content leak out, is going to injure and destroy the nearby tissue. It's going to injure and destroy the nearby tissue. Yes. The haploid sex cells? they are not in a state of G0. They are waiting for the other haploid cells so they can combine together and both become diploid. And then there are proteins, transcription factors that will initiate transcription, and then you'll have the proteins that will push the cell into the cell cycle. So they will divide into gastrulation, into blastula, into an organism. Does that make sense? What is your question? I don't get your question.
If we have to say what phase a cell is in, I understand it from mitosis, meiosis. So I was like thinking, is it the endocytosis or is it meiosis?
So once you leave mitosis, even meiosis is just like mitosis, but meiosis for sex cells. This is mitosis. So sex cells, they cannot be in a state, we cannot look at them in a stage of terminally differentiated, because those cells, once they get fertilized, they start dividing. So we're not talking about reproduction. That's reproduction. This is here for healing. So for healing, you are examining the capacity of the tissue to regenerate or not. Regenerate means it can re-enter the cell cycle and it can be able to heal. But you don't heal reproductive cells, sex cells, because they are not there as a tissue. They are there for potential, to potentially get fertilized and then break apart and give me, they blast off and they start making embryological development and turning into like an organism. Okay? Yeah, part of it, but it is a distinct phase. Like this is just some extra information that you really don't have to worry about. Like I mean, you don't have to go into the detail of when exactly does it begin, but 80% of it is distinct. But there's some gray area. There's some gray area where cytokinesis begins just a little bit with anaphase, because with in anaphase you don't just pluck off the two sister chromatids. Some of the cytoplasm starts to shift, but all you need to know for like this purpose, like NCLEX and all that stuff, just know the stages that they flow in that order. But they're not gonna ask you a question like, when exactly does cytokinesis begin? Does it begin as a distinct phase or does it overlap with the one before it? You know what I mean? But sometimes some professors, they just like to add some like extracellular information, extracurricular information. Anyways, so but now we understand this is the problem here. That programmed cell death, we need that to remove the cell properly. But when we get older, that apoptosis is expensive because it's helpful. Dividing the big cell into plebs, that requires a lot of energy, requires a lot of oxygen. That's why people who have heart attack, that's death by necrosis, because there was no oxygen. There's no oxygen. The mitochondria says, give me oxygen so I can do programmed cell death. The body would say, if there was oxygen, you wouldn't even need to do the programmed cell death. We don't need you to kill the cell, but there's no oxygen for nothing. So the cells are going to start rupturing. They're going to die by necrosis, and all the content is going to leak out. It's going to leak out. The lysosomal content is going to leak out and is going to destroy the nearby tissue. It's going to destroy the nearby tissue. Okay. Replacement with dense fibrous mass. Okay, so what happens, that regeneration here, when we heal by entering the cell cycle and rebirthing and having more tissue, that is known as regeneration. That is regeneration. That is regeneration. Okay. The scar is a fibrous tissue formed during healing and may differ from the original tissue. Okay. Replacement with dense fibrous mass. So what happens now if you are a terminally, so maybe Tenzin, maybe you're talking about What if a cell that is cannot enter the cell cycle, that is non-regenerative? These are non-regenerative cells. These non-regenerative cells, they get damaged too, like your nerve tissue. We talked about that. The nervous tissue in Alzheimer's disease or in dementia, when it gets damaged, it cannot heal by regeneration because it cannot enter the cell cycle. It is these epithelial cells, they can enter the cell cycle and they can spin the cell cycle. So they can heal by regeneration, but this tissue cannot. So this tissue that cannot heal by regeneration, it heals by scarring, heals by scarring, also known as scar tissue formation, scar tissue formation, scar tissue formation. And this scar tissue formation, this scar tissue formation is a form of a tissue that is formed because there's fibroblast cells. Fibroblasts, they initiate TGF beta. These TGF beta cells, they are the ones who dump a lot of fiber in the place where there is injury. So when you carve out and you remove all the necrotic debris from the tissue, that's the tissue, and you have removed the damaged tissue, but the tissue is non-regenerative, it's quiescent. It cannot rebuild again, then it's rebuild by going to rebuild by dumping a lot of scar tissue like cement, and that is going to be fiber. That's known as healing by fibrosis. Healing by fibrosis. Healing, healing by fibrosis. Healing by fibrosis. Replacement with dense fibrous mass. These are sometimes damaged tissue that is replaced with dense scar tissue. A keloid is an overgrowth of scar tissue extending beyond the wound site, and that is often difficult to treat. There is a keloid, and there's what's also known as hypertrophic scar. Hypertrophic scar is a scar that has healed but is hypertrophied. It went far beyond the boundaries. And also the keloid went far beyond the boundaries, the outlines. So you would see like a big blob of like scar tissue that is dangling from the like region where the patient is injured. You see, this is the healing of a minor wound. So the blood clots quickly, stopping blood loss. And the germinativum cells grow laterally to close the gap and restore the epidermis. And the clot dissolves, leaving repaired epidermis and thickened dermis. So the body membranes, these are thin sheet-like structures. These membranes covering and protecting the body surfaces and organs. So many important functions, including the barrier protection, the substance exchange, and fluid secretion. So there are two major types. There are epithelial membranes and connective tissue membranes. These epithelial membranes are made up of epithelial cells and are underlying layer of connective tissue. They secrete mucus and other substances to protect and lubricate the surfaces, and they aid in the absorption and the secretion processes. The connective tissue membranes, they are composed of connective tissue only. Unlike the epithelial membranes, these are made up of epithelial cells and an underlying layer of connective tissue, and they secrete mucus and other substances to protect and lubricate the surfaces. The connective tissue membranes, they are composed of connective tissue only, and these membranes, they secrete a watery fluid called serous fluid, which lubricates the surfaces of organs and reduces the friction during the movement within the body cavities. These are the types of body membranes. See, that's the mucus layer, that's the epithelium, and these are the goblet cells. Okay, epithelial membranes are represented by the cutaneous membrane, the serous membrane, secretes fluid, and it lines the cavities and is not open to the external environment. The mucous membranes, known as the mucosa, they produce mucus, such as cilia, and they line the body cavities, opening directly to the external environment, and they have a role in the immunity. Okay, we have synovial membranes, and these are the membranes of the connective tissue that is responsible for providing tensile support and integrity to the bone structure. Okay, so mechanisms of disease represent this stage and beyond. So when we heal, so now when we go through scar tissue formation, this process, so right after we take a detour, so right after necrosis, there is a spillage. So the nuclear plants spill to the neighborhood that is nearby. So all the houses are damaged. Same thing with a cell that is harmed by necrosis. All of the membranes are broken and all of the lysozymes, the degradative enzymes, the metalloproteases are leaking out and destroying the nearby tissue. This nearby tissue is now harmed by the own substance that is inside the cells. So now what happens is your body must enter into a state known as inflammation. And it has actually two stages. There is acute inflammation, and if it is rectified, we go to healing by regeneration or healing by regeneration or healing by first intention. But if the damage is so severe and the duration of the insult is so long, and if the onset was so sudden and rapid, the intensity was so severe, the duration was so long, then we progress from acute inflammation into chronic inflammation, And this chronic inflammation is able to more aggressively attack the site where you have necrotic debris, where you have infectious microbes that are infesting the tissue, and then it will fight and combat and remove all of that dead tissue and all of those microbes, and then it will start the process of healing by regeneration, provided that the tissue that is healing is regenerative tissue that can enter the cell division cycle. But if the tissue cannot enter the cell division cycle, it will be healing by fibrosis, will be healing by scar tissue formation. And what mediates inflammation? There are 5 cardinal signs of inflammation. The first one is redness. So redness is known in Latin as rubor, and there's heat known as calor, and there is pain known as dolor, and there is also swelling known as tumor, and there is loss of function known as functiolasa. These are known as 1, 2, 3, 4, 5 cardinal signs of inflammation. So an inflamed tissue will suffer from all 5. It will be red, be pain, will be swelling, and there will be loss of function, and the area will be hot, be more hot than the rest of the body. So what is causing that? What is causing that is that it began with a cell injury and cell death by necrosis. We're never going to have inflammation if we die by apoptosis, because the cell has now been broken down into edible blebs. Blebs are harmless blebs. It is the necrosis where all the lysosomal enzymes, the degradative
proteolytic enzymes are leaked out into the tissue and they destroy the nearby tissue, and then you will have redness, heat, pain, swelling, and loss of function. Swelling and loss of function. So for that, you would have a lot of extra blood supply. So if you have a neighborhood where there is gun violence or gang violence, you will get a lot of cops running to that neighborhood. They're not going to be running to a neighborhood where it's safe. So the same thing, a tissue that is inflamed with a lot of is infected with a lot of microbes, you will see the blood rushing there with a lot of white blood cells and immune cells to remove all of that necrotic debris and fight against all of the infectious microbes. So when the blood is running there, it's going to be more red because the blood is running there. Also, it's going to be more hot because there's extensive movement of blood to that direction. If you have a river that's flowing and you have a lake that's stagnant, you put a thermometer in the stagnant lake and you put a thermometer in the running river, which one is more hot? Sorry? The running river. So the same thing, the area that has a lot of inflammation and a lot of blood moving to that area is going to be more hot. That's why there's heat. There is pain because inflammation is mediated by mediators, known as the mediators of inflammation. These mediators of inflammation, they irritate the nerve endings. They irritate the nerve endings. And when they irritate the nerve endings, you feel pain. That's why with inflammation, there's a lot of pain. And then there's swelling. Why? Because a lot of blood is arriving to the tissue, and that leads to the formation of an exudate. The exudate means that there's a lot of water within the blood is leaking out, seeping the water, seeping out from the injured tissue and from the blood vasculature into the extracellular space. And that causes the bump swelling. And functional lesa, meaning loss of function, so the tissue that is hot, pain, inflamed, is not going to be able to do its function. If you cut your hand, you're not going to be able to use it. You're going to be able to use the other one. You're going to lose function. So if you have these 5 signs, these are the signs of your inflammation. So not all diseases are inflammatory diseases, like cancer. Cancer is painless. If a patient has cancer, they don't have pain, they don't have heat, they don't have swelling in the beginning. So cancer is uncontrollable cell division because cells are dividing and dividing. It is in the later stages where the cancer cells have detached from their original location, and they have invaded nearby tissue, and they have sneaked into the blood, and they travel in the blood from the bone cancer. If they travel in the blood, they reach the lung, and then they attack the lung. That's metastasis. When they attack the lung, they start infiltrating to the nearby tissue, destroying tissue, then that inflammation. Then the patient starts feeling pain, but it's too late. Now the disease transitioned from cancer into inflammatory disease. But in the beginning, it was not inflammatory. It started as cancer, but it became inflammatory. Sometimes we have the opposite, like the human papillomavirus. That's not cancer. That's inflammation. That's a microbe. That's virus. So the HPV, the human papillomavirus, that's the infectious, like the microbe. You see, I put here infectious microbes. So infectious microbes are the cause of inflammation. So human papillomavirus start with inflammation, but then it develops into cancer. So they can cross road. You can begin with cancer, end up with inflammation, or begin with human papillomaviral infection, end up with cancer. What is the third type of disease? That is neither. There's a third type of disease that is not inflammation, that is not cancer. What is that? So you can only have 2 types of disease, cancer or inflammation. What's the third one? Jeffrey, would you like to buy a bio? Did they do that in that one? That's the other one. Spinning the wheel. That was wheel of fortune. Okay. What is the third type of disease? Congenital disease. You are born blind. Is that cancer? That pain, you're just born. That's an embryological defect. Or you're born with a horseshoe kidney. They are your kidneys are attached together like a horse, like a horseshoe in the bottom of it. So the kidney does not look like that. Not look like that. The kidney looks like this now. So these are known as congenital disease. These are diseases that you are born with. Congenital disease. These are diseases that you're born with. Okay. Somebody emailed me about breaks and all of that. Okay, so I record the lecture. Anytime you feel like you want to take a break, you can step out and it's recorded. You're not going to miss anything. And at home, you can check it online. And the part that for which you have went out, you can just find that part in the recording. I don't like to decrease learning time. I want to like give as much explanation, as much wealth of information as you can get. But and that's why I record the lecture. You can go home and review it there. But yeah, if you want to, at any time you may leave. And since last week we didn't give you a break, you didn't know you could leave the room. Then today I will, since I owe you from last week, so I'll dismiss you. I also want to give you time to work on. Did you raise your hand if you finish your study guide? And that's not a good rate because there's 100 questions in there. I guarantee, I think more, like 100, maybe 110, but they're all based on the study guide. So it's a double-edged sword. It's either that day, it's either you're getting 100 or you're getting a 0, because it is, and no rebound from that. You get a 0, no rebound from that. You can do all the homework questions. The homework questions, they are like ratio. They are a ratio. So if you get 100 on all the homeworks, okay, then they're going to be reduced into your homeworks that you think they worth how much, 10% or 20%? Yeah, 10%. So these homework questions are also go with the study guide. You saw that, right? You saw that the study guide question and the homework questions and the lecture content, they are going in the same direction. You're not like all over the place. So make sure that you strategize, because yeah, it's there. The exam is like an open book, but it's not actually an open book. You have to already solve it from before you come that day. You just pop, 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 you choose the answers. So you have to finish it and upload it the night before. It is unaccepted if that deadline is irreversible. So please make sure you go, you solve your study guide, and make sure you do your homework. And these homework questions, make sure you study every single homework question and understand why is everyone right, why is everyone wrong. Because these homework questions, I simulated them. I simulated them for your exam, and they're going to help you. They're going to help you a lot. They're going to help you a lot to